In this video, we are going to discuss about blotting in detail, its types and its procedure in detail. Blotting is defined as the transfer of biomolecules like DNA, RNA, proteins, and other modified biomolecules from an electrophoretic gel to a membrane for further analysis. It can be applicable in many fields like understanding the gene expression by analyzing the messenger RNA, genetic diagnosis by studying the mutations, and also we can interpret protein expression and function. There are four different types of blotting. The first one is the southern to analyze the DNA, then the northern used for analyzing the RNA sequences, the western is used for analyzing the protein structures, and lastly, the eastern blotting is used to study the genetic modifications. In all types of blotting, the analyte is transferred from the gel to a permanent membrane. The purpose behind the transferring of analyte on the membrane is that membrane provides favorable environment. The membrane provides analyte to be accessible. It is easier to detect. The gel is fragile, more prone to break. The membrane is more durable and easy to handle. The membrane can be repropped multiple times. It also reduces the background noise and it has increased the sensitivity and specificity for the. Now we will discuss Western blotting in detail. The steps are pre-blotting, gel electrophoresis, blotting, and antibody incubation and detection. Pre-blotting. In this step, the sample is prepared, a specific protein is extracted by cell lysis and centrifugation, and its concentration is quantified, and the sample is ready to be loaded onto the polyacrylamide gel. The next step is the gel electrophoresis, in which we apply current and analyze the gel, in which the proteins start separating on the base of their relative molecular size. The third step is the blotting, that is the actual transfer. For this, we assemble our transfer buffer, the gel, and the membrane on which we are going to transfer our molecules. Then, the proteins from the gel are transferred to a nitrocellulose or polyvilidene fluoride membrane by various methods that could be as followed. The transfer of molecules can be done by electroblotting, that is by providing current, and by capillary transfer, that is by use of the filter papers, and it can also be done by vacuum transfer, that is by creating a vacuum in the chamber. The transferring time for the binding of the proteins on the membrane can be dependent on two factors, that could be the size of the analyte, that could be small, medium, or large, and the second one is the type of the membrane we are using, that could be the nitrocellulose membrane or the PVDF membrane. The use of BSA, bovine serum albumin, is used to block the proteins on the membrane by incubating in BSA buffer to prevent non-specific binding and washing out from the membrane to minimize the background noise. The fourth step of Western blotting is the detection, for which we first of all use the primary antibodies that are specific to the targeted analyte and added on the membrane and incubated, and the excess is then washed off. Then we use the secondary antibodies that are specific to the primary antibodies and are labeled. Then the excess secondary antibodies are incubated and washed off. The secondary antibody can be labeled in many ways, such that by the use of enzymes by converting a colorless to a colorful product, by using fluorescent molecules that fluorescence, and by using a chemical that gives a colored product, or by using a radioisotope that emits light on exposing to X-rays. The last step of the Western blotting is the analysis, in which we analyze the image derived from the results of the blotting assay, that could be in form of a colored product, and we record the results in a form of paper or in our computers. Same steps are followed in all types of blotting, just the detection method changes, like the use of probes that are complemented to the targeted DNA or RNA in northern and southern blotting. In conclusion, the blotting test gives more quantified information about the analyte or occurrence of any disease in a sample or a patient. At the end, thank you so much for your attention. Hope you understand the concept of the blotting, and good luck. At the end, a special thanks to my fellows without whom the video could not be possible.